fans.
Microscopy 2, Bio 100, prepared by Miss Celestial, Miss Alpides, Miss Galvante, Miss Ramos, and Miss Santo Domingo. Introduction. In this activity, we focus on examining the cellular features of onion, tomato, and pigment samples using a compound microscope. Learning objectives. To observe and identify the structural characteristics of onion, tomato, and pigment, and to develop proficiency in adjusting light, focus, and contrast to produce clear and accurate microscopic images.
Good day, I am May Divine M. Celestial. Today, I will be explaining the methodology used for our microscopy activity. First, the procedure begins with wearing proper laboratory PPE such as a laboratory gown, gloves, and a face mask to ensure safety while handling the materials and specimens. Next, the materials and samples are prepared, including onion, tomato, hydria leaf, and squash stem. Each specimen must be clean and ready for slicing. After preparation, thin pieces of the epidermis are sliced from the onion bulb and the ripe tomato. Thin slices allow more light to pass through, resulting in a clearer observation under the microscope. Then, each specimen is placed on its own glass slide to prevent mixing of samples. Once the specimen is positioned on the slide, a drop of iodine solution is added. Iodine serves as a stain that enhances the visibility of cell structures. After adding iodine, a cover slip is gently placed over the specimens to protect it and flatten the sample for easier viewing. The prepared slide is then placed on the microscope stage, starting with the scanning objective, followed by the lower power objective, and finally the high power objective for clearer and more detailed observation of a cell structure. Lastly, observations are recorded for each slide, noting the shape, colors, and other visible cell details. This concludes the methodology for the microscopy experiments.
My name is Pia Santo Domingo, and now we'll come to the result in discussion. So, the first slide is the onion. The scientific name is the Allium cepa. In the scanner, we observe that it has rectangular, brick-like, and arranged in a very organized, parallel pattern. The tissue is stained purple or pink, making cell walls highly visible. The surface appears smooth and continuous with very little tearing. To the LPO, distinctively rectangular, arranged in a straight, brick-like pattern. Cells are aligned in parallel rows, which is characteristic of epidermal tissue from onion bulb scales. The HPO is the elongated, rectangular cells arranged and not rows. Pinkish stain, which enhances visibility. Clearly visible cell walls, thick and wavy due to membrane shrinkage or lighting. The onion epidermis is a single layer of parenchyma cells. Next is the tomato or Solanum lycopersicum. The scanner we observe here is that the specimen appears as a thin, yellow-colored sheet of tissue. No visible chloroplast. Surface appears smooth and uniform. Its scattered dark circular or irregular spots appear across the sample, likely air bubbles or debris. To the LPO, the epidermis of tomato appear tightly packed with a polygonal shape and clear cell walls visible. The overall coloring is yellowish, indicating natural pigment effect. To the HPO, it is polygonal or rounded cells, thin and clear cell walls. The color is from a tomato pigment, usually carotenoids, giving the yellow-orange look in microscope when the light passes through.
Hello, I'm Marian Jane F. Galvante, and I'm here to discuss the results and discussion for the digman, also known as Hydrilla verticillata. Sa scanner, we can see thin green leaf-like tissue with elongated rectangular cells. Cell arrangement is more elongated and less uniform than onion cells. Some jagged or irregular edges appear, suggesting torn tissue. Circular bubbles are visible near the edges of the sample. For the LPO, we can see rectangular and arranged in a uniform grid-like pattern, and the image shows transparent cell walls and a structure suggestive of a leaf epidermal cells, possibly from a monocot plant. In the HPO, we can see elongated brick-like cells, greenish to yellowish coloration from chloroplasts, cells arranged in rows typical of palisade mesophyll, and numerous chloroplasts giving the tissue a green and yellow tint. So in this activity, we examined an onion root tip under 3 different magnifications: the scanner, low power objective, and high power objective, with the goal of observing mitosis. Typically, onion root tips are excellent samples for viewing the stages of cell division because the meristem contains many actively dividing cells, but unfortunately, in this sample, we didn't see the mitosis. Sa scanner view, at the lowest magnification, we can only see the overall structure of the root tip. The tissue appeared intact, but no distinct cellular details were visible. Since mitotic stages require individual cells to be seen clearly, this magnification did not reveal any signs of cell division. For the low power objective, at medium magnification, individual cells began to appear as columns of rectangular structures. Even at this magnification, none of the cells show the distinctive features of mitosis, such as condensed chromosomes, spindle fibers, or a dividing nuclei. The cells appeared elongated and uniform, suggesting they were in interphase, the non-dividing stage. Lastly, at the high power normally allows us to see mitotic phases like prophase, metaphase, anaphase, and telophase. However, in our slide, the cells showed no distinct chromosome shapes or division patterns. Instead, the cells appeared lightly stained and uniform, with no signs of chromatic condensation. This means the cells were likely not actively dividing at the time the root was preserved.
Good day, I am Catherine Faith Alcadas, and this is the conclusion of our activity. The activity used the scanner, LPO, and HPO lenses to observe prepared slides of onion, tomato, digman, and squash. Each objective showed different level of details, helping reveal the basic structure of the plant cells. Proper slide preparation made the samples clearer and easier to view under the microscope. Sharing observation, working together, made the process smoother and more accurate. Overall, the activity improved microscopy skills and understanding of plant cell structure.